So we'll first start with Dr. Marques from SOPHiA GENETICS. So Dr. Marques has a rich career in biology with strong expertise in genomics. She's now the head of knowledge transfer in SOPHiA GENETICS. And without further hesitation, I invite you to join us on the stage.
Thank you. This is my pointer. Okay. So thank you, and first of all, let me start by congratulating the organization for an amazing event. I think we had through the day a lot of different perspectives and different paths that are available to scientists in industry. And so what I'll try to do in the next 20 minutes or so is to try to give you a flavor of what is my path and how it led me to be transfer knowledge. And what I hope I will be able to allow you to understand is this picture by the end of the talk. So how did the career path that I've taken has led me to this place where I organize or co-organize, among other things, events. This is the annual meeting that we have at SOPHiA, where we discuss with the teams roadmap and why is it acceptable that I co-host this wearing a DIY astronaut costume. So, where it all started. It's not as beautiful as Yemen. I must admit that I was really tempted to visit after your talk, but it's very colorful and bright. And so I was born in Lisbon and I grew up in Sintra, so I don't know if any of you know the region, but if you don't, I really encourage you. It's a beautiful city, both Lisbon and Sintra. So this is where I grew up. And I think I might disappoint some of you, and I hope you don't think I'm not cool after what I'm going to say, but as growing up, I loved school. I was really one of those kids that really, I loved school. I loved learning, and I loved everything about school. I really loved science, but I also enjoyed quite a lot writing and reading. And so for a long time, and I think it's because I was growing up during the Gulf War, that was very formative. I wanted to be a war correspondent. I really wanted to be a war correspondent. I wanted to be a journalist. But at some point when I was growing up, when I was growing up, somewhere when I was, I think, in middle school, I learned about DNA. And I think we heard this morning on how some of these lectures can be very formative. And actually, this fascinated me. This idea that, this is an old version of the talk, but it's okay. So this idea that all of the information that was contained and was necessary to make a human being was, as well as animals, was contained in our DNA. And actually, probably the idea of the dinosaurs is really to illustrate that not only was this information important to determine the different traits that distinguish between us and to understand some aspects of disease, but actually it also encoded some of the information about human history as well as the history of other living beings. So this was very amazing to me at the time, but growing up, really being a scientist was not something that I understood as being a job. And so I didn't really knew that you could be a scientist, except if you were a Hollywood actor. So I kind of had to concede into going and do something that was somewhat more acceptable. And because I was reasonably good in math and chemistry, and biotechnology was something that had to do with DNA, and I really, every time I spoke to people that did biology, they told me that I was going to be a school teacher, and honestly, I'm not very patient, and I don't think I would do a very good job. I've kind of settled on this path. So I did a 5-year degree on chemical engineering. So this was before the old days, so it was like a long kind of bachelor and master together. And at the end, I was a chemical engineer. So that was okay. I didn't particularly love it. I didn't particularly hate it. But I think what I really learned when I was in this very good school was that actually there was people hanging around in the labs that were scientists. They were doing experiments. They weren't doing the type of experiments that I was interested in, but they were doing stuff that looked cool. And so this seemed to be a job. And so that made me understand that there was a career path that was possible through science. But I really didn't want to do research in chemistry. I wanted to kind of go back to my first love. And so when I finished my diploma, basically the day I finished, I looked at my parents and I said, I've done this. Now I do what I want. Even though they never really forced me to do chemical engineering, but it sounded cool. So I decided that I was really going to try to find my path. And this took me to about 9 months of really trying to understand how I could navigate my way back into biology. So for 9 months, I was working during the day in a clinical diagnostics lab, so kind of analyzing people's blood. That was interesting. It gave me a lot of insights about how you do experiments, how you manage kind of large volumes of experiments, because basically that's what you have to do. And then in the night or in the evening, to be more appropriate, I was doing this internship for free in this foundation, Gulbenkian for Science. So this is a very, I think, somewhat internationally renowned foundation near Lisbon. There is a lot of good scientists, a very large and broad community. And there was one group of old physicists. They were physicists that had converted into evolutionary biologists that were happy to take me on board. And so for 9 months, I was doing experiments, learning about evolution, and I learned quite a lot over those 9 months on how do bacteria evolve antibiotic resistance. And so I learned a lot about evolution, and it also kind of provided me opportunities to understand how I could apply for a PhD, because really I also at that point understood that if I wanted to be a scientist, probably the easiest way was to get a PhD. And so I've applied for a number of different PhDs, and so I ended up being lucky, if you wish, and I kind of got the chance to do a PhD really on what was really the thing that fascinated me, and that was to try to understand what makes us humans from the molecular perspective. And so I did a PhD at the University of Lausanne with Professor Kessman when he was just joining the CIG, which is not very far away from here, on the role of gene duplication in the origin of human genes. And so those are amazing 5 years. I actually had a lot of fun. I've did a lot of experiments, learned a lot about molecular evolution, and I also got the chance to work quite closely with the bioinformaticians. And I realized about towards the second half of my PhD that if I wanted to really understand what makes us humans and to kind of really leverage on all of the data that we were generating through these new sequencing technologies, actually pipetting might not cut it. So I really needed to kind of build up my computational skills and be able so that I could really take advantage of the data. I think this was about, I think I remember seeing the publication for next generation sequencing kind of halfway through my PhD, so you can see how old I am. And so this was really, the field was really booming. And so I finished my PhD and things went well, and so I had two missions. And I think this is, people asked about how you navigate changes in a personal life, and so I'll give you a little bit of how some of these personal decisions also influence some of my choices. And so I wanted to learn how to code. That was one thing. But in the meantime, I had also found who is now my husband, who is a nuclear physicist. He worked just not very far away from here, and he had then moved to Oxford to work in this big tokamak experiment. If you understand nuclear physics, you know that this type of engine is not something that there is 100 in the world. So he was bound to Oxford at the time, and basically I kind of needed to find a way of going. And so I found some people that were willing to host me, but they didn't really have the jobs, and I'm not very good at waiting. And so I decided, well, I'll just go and find some money. And I was very lucky because I had done a very good PhD. I had a decent PhD, so I was very lucky to be funded throughout my entire postdoc. So I initially got funding from the Swiss National Foundation to move to Oxford, and I kind of got some money from the Portuguese National Foundation, and the last 2 years of my postdoc were funded by a Marie Curie Fellowship. And so I've learned a lot during my postdoc, but I kind of would say that there is four take-home things that I would like to bring. So first, I think being able to combine all of these different approaches, so the molecular biology, the chemistry, the clinical diagnostics with the coding, actually ended up opening a lot of doors. I had a very large toolkit that I could use, which made me somewhat attractive to people to work with. It also made me more easily pick up on different topics. Because I had my individual funding, I know this is not the case in all of the labs, but in the lab where I was, I actually had quite a lot of freedom. So I was not, well, I still needed to work on things that were relevant to the rest of the lab, because otherwise it wouldn't make a lot of sense. But I could choose with whom I would collaborate, I could choose the projects that I would do, and that gave me a lot of understanding on how interesting it is to work as part of multidisciplinary teams, and I think getting my own funding kind of gave me that opportunity. I've mentioned that I wanted to be a journalist, but actually by the end of my PhD, my writing was not crap, but it was not great. And so I think one thing that I've learned a lot during my period as a postdoc was really how to write, and I got that by practicing. So practice makes it perfect. So I took all opportunities to learn how to write, kind of reviewed papers for everyone in the group, discussed my own papers with people. So this was something that really helped. And I think this was touched upon early today. And I think it also made me realize the value of really working with people that really inspire you. So my mentor, my postdoc mentor, is someone that really inspired me, that saw potential in me that I didn't see, and kind of provided me with all of these opportunities. So I think if you can choose someone that you can work with that inspires you and that will serve you for life, and then again, whenever you have the chance, you should give it back 2 or 3 times. Okay, so my postdoc went well, and at the end of it, after like about 3 years, I was getting to this point where I was having more ideas than I actually had hands. And then my postdoc was like, my postdoc mentor was like, I think it's time for you to fly the nest. And so that led me to really kind of developing my own research program that I initially started in the context of I was in part of the same department, so I was a research fellow in the same department as my mentor. But obviously, as you mentioned, this was never a very good idea. So this was, again, funded on soft money, and what we were working on was non-coding RNAs, and so I think I don't need to tell you very much because Dimitri had already given a really good introduction, but for those of you that were not here, I'll just recap. So if you remember about what I told you about what was my interest, is really genotype to phenotype, and by the time I started my postdoc, it was clear that most of our genome is non-coding. Well, it had been clear for a while, but probably more exciting that a large portion of it was transcribed. The thing is that we really didn't know the functions of most of these things. I don't have my count stopped about in 2020, so I don't know how much this percentage has increased. I think by the time was about 2%. But what we knew was, as Dimitri said, is that as a group, these molecules were very versatile and that they could perform all of these different functions. And so my idea was that if we were able to understand what were the molecular features of these functions, we would understand the code, and similar to proteins, we'd start be able to predict what these things do, because it was starting to get, at least towards the end of my life in non-coding RNAs, rather frustrating to say we have all of these things that do all of these amazing things, but we don't know what they are. And so that was my idea. And so, as I said, I was pushed out of the nest, and it was time to move. And at this time, we were no longer 2, we were 3.5, and we were kind of starting to think that it was time to come back to Switzerland. And as I told you, I'm not very good at waiting, so I thought, well, I'll do the same thing that I've done again. And so I came back as an SNF professor, even though many people told me that this might not be the best idea. I came back as an SNF professor. I was initially hosted at the Department of Physiology, but spent most of my professorship at the Department of Computational Biology, and then kind of supplemented my professorship with a number of different trainings. And so the slide the image of the papers is just to kind of give you. No, it doesn't cover all of the publications. We published more. In a way, I wanted to be reassured that I was just not fuffling around, that we actually did some work, but I think all of these publications somewhat illustrate some of the things that we were working on. We were working on long non-coding RNAs, their contributions to disease, and the molecular mechanisms that might underpin them, as well as on microRNAs. So this was kind of going okay. The group was never very big. I think we were at most 4. We were having fun. But I had this non-tenure position, right? So if you come with an SNF professorship, you have 6 years to find a job. And so at some point, I really needed to find a job if I wanted to stay in academia. So that was quite a lot of energy that was invested into this. And so this is kind of a warning, or kind of, and I know not a warning because I think it's okay, it makes sense, but I think I at least was not aware that this was going to happen. And so I'm just basically saying it to everyone. So I spent about 18 months of my professorship with really my only focus was, where do we go next? And I think what was daunting at the time is that we were a group, so it was not only me as a postdoc, I had all of these people that were relying on me. So I think I've counted, I went through my application folder, and so I've done about 30 applications, which is not too bad, because I was restricted to Europe. I've decided that I was not going to go everywhere. And I interviewed in 10 places. And actually, what this time was interesting for was actually it gave me a lot of opportunities to think about what I liked, what I didn't like, what I liked about academia, what I didn't like. And at the end, the outcome was positive, so I got a position. So I got a position, it was a permanent position, so a 10-year permanent position, associate professor position with research money and not that much teaching, so it was not too bad. It was just that it was not in Switzerland. It was in the UK, and it was okay, we had lived in the UK. And so I accepted this position, and I had it about 6 months to do the transition. And so over those 6 months, it was very tough, because I just was not sure I wanted that position. And it really made me think a lot about what was the underpinnings of that decision, right? I had the time to think, and I thought, well, really what I care when I go to work is really those things that I'm listing here. I love being part of a team. I love solving problems. I love learning new stuff. So I'm still kind of a school geek. You will see in a minute that's true. I love genomics, and I really like writing. And actually, I like storytelling in general. And I think for once first time in my life, as in my adult life, I really liked the feeling of feeling at home. So I was no longer prepared to leave. And so I think after a lot of thought, I kind of thought, well, potentially all of the things that I really like doing don't really require me to stay in academia. And so I kind of really literally opened a browser, and I ended up on this job description. It's not fancy. It was not a fancy description. Sorry, Jean-Rénia. It was basically a position, it was a senior scientist position. I don't remember the details, but it was a lot about project delivery and external and peer review publication, which I like. Oh, I like writing, I like handling projects, and I'm senior enough to be a senior scientist, so why not? So I applied. So I think, oh, I'm missing the slide about SOPHiA. So I don't know if you guys know what SOPHiA Genetics does, but SOPHiA Genetics, at least for the genomics community in Lausanne, is very well known, very well established in the environment. It's a platform as a service. Is kind of the key product is the SOPHiA DDM, and this platform enables clinicians to analyze and extract insights from their digital health. So this sounded cool. So I applied. And somewhat they thought I was good enough, and then they gave me the job. And it was very immediate. In contrast with my academic job, I was really immediately very excited to join the company. I think I've met a lot of people that were fun, and I kind of also heard a lot of interesting things about how in industry work, a lot of the focus is on the we and less on the me. And so a lot of those things resonated with me. So I've accepted the position, so I rejected my position as an academic, and I started on the 1st of April 2020. It was very fun. You might remember things shut down 2 weeks before. I got my computer at home, and I was sitting there kind of thinking, how am I going to manage closing my lab? Because actually for the first 6 months of my tenure with SOPHiA, I was working part-time because there was an agreement, and that I think it's important to highlight that. I think sometimes we hear that companies are not flexible. I think this is an example of flexibility. They really wanted to ease my transition and to make sure that I fulfilled my commitments. So they allowed me to work part-time for the first 6 months, which was not a bad thing because anyway, no one was working, right? And then I onboarded 100% in September. So that's how it started. I started as a senior scientist. And what do I do now? So now my job as a head of knowledge transfer really involves any activities that relate to disseminating the knowledge that we generate as part of the R&D teams. So at SOPHiA Genetics, R&D is about 46% of our workforce. There is a lot of R&D development, so the role of my team is really to disseminate that. So we do it through publication writing. We write external publications, peer-reviewed, as I used to do. So this is directed to externals. We also do a lot of technical writing. Well, a part of the team does technical writing. And what technical writing implies is really writing the good protocols that the users can use, as well as making sure that the ways we communicate or that we document our development activities are compliant, but also easy to use for the teams. So that's what the technical writing team does. I have a stint on intellectual property, and this is really interesting, and I wanted to highlight that because I really didn't had any experience with intellectual property, a management of formal intellectual property assets before I joined SOPHiA Genetics, but I was kind of given the opportunity to onboard the IP team and kind of increased my contribution there. So now I currently manage the patent portfolio with the support of our councils, and I'm also doing a master in IP management and law really to support this activity, so really allowing me to continue to grow despite the fact that I'm starting to be quite a senior scientist by now. And so the last thing that my team does is training. So we handle part of the training, so not all of the training in the company, but the training that pertains to technical activities. So making sure that we upskill both our sales teams as well as our dev teams to basically adopt the processes and understand the products. And this is how I ended up here. So one of the things that I really have the chance, and really I think it's a privilege to do, is to co-host with the sales team our annual kickoff. And this is really an important opportunity because it's the time of the year where we bring everyone across the company, and the workforce is located worldwide, and we need to basically engage everyone and motivate them to think of what is the focus for the year to come and what are the things they should be focusing. So it really requires a lot of work, so that's why I have a lot of appreciation for what has been done here, but it also involves quite a lot of creativity, and we thankfully have the chance to be allowed to be creative. And we are very proud of our toilet paper astronaut helmets that you can see there on the bottom. And with this, I would like to thank you, and I'm happy to take any questions after that. Thank you.
Thank you very much. So do not forget to submit your question online.